Hello everyone, my name is Nour GHAZZAUI, postdoc at Cribble Laboratory Limoges. I work with Dr. Denizo's team on a project investigating the role of the two major cis-transcriptional enhancers of the IGH locus on class switch recombination breaks reparation. Mature B cells express on their surface immunoglobulins, which are constituted of two identical light chains and two identical heavy chains. Heavy chains are encoded by the immunoglobulin heavy chain locus, known as IGH locus, which is composed of a variable domain leading to the antigen binding site and a constant one containing the different genes encoding the different immunoglobulin isotypes. To be functional, this locus must undergo a series of rearrangement where it needs to break its DNA, resulting in the insertion of an oncogene into the IGH locus, which is the cause of many B-cell lymphoma. This is the case of BCL2 translocation in follicular lymphoma taking place during VDG recombination, which is an essential event to generating a functional immunoglobulin repertoire. The translocation of c-MYC in Burkitt lymphoma taking place during somatic hypermutations, which is essential to affinity maturation, and the translocation of cyclin D1, D3, CMAF in myeloma taking place during class switch recombination event, which is important to change the immunoglobulin isotype here from an IgM toward an IgG1, for example. So, these chromosomal breaks are under the dependence of the two major cis-transcriptional enhancers of the IGH locus, known as EMU, which is known to control VDG recombination, and the 3' regulatory region, or simply 3' RR, where we have already shown that it controls both SHM and CSR. So they are important for DNA breaks. But what about their role in its reparation? Do they participate in DNA reparation and therefore possibly in the insertion of the oncogene into the IGH locus? For this, we analyzed junctions resulting from CSR mechanism, which is a two-step process. The first one consists about the induction of DNA double-strand breaks, and the second step consists about their reparation by joining the most switch donor region, usually switch mu, and one of the downstream S acceptor region, which is in our case, switch gamma 1. This reparation phase is carried by the non-homologous end joining pathway, which promotes blunt and microhomology junction in favor of the alternative end joining one, which promotes large homology structures. To study the impact of these enhancers on the second step or CSR, and because there's a strong homology between IGH locus in human and mouse, we sequenced the junctions resulting from the culture of splenic purified B cells of white type, 3' RR deficient and immune deficient mice models. These cells were cultured for 4 days with different cytokines cocktail in order to induce the switch toward IgD3, IgG1, IgA. DNA was extracted, libraries were prepared for sequencing. The analysis of breaks reparation profile showed no effect of the deletion of EMU enhancer on the junction profiles on all of the 3 different isotypes, IgG1, IgG3, IgA, and all of the different structures, blunt, insertion, microhomology, and large homology. While it's not the case in 3' RR deficient mice, where there's an impact on IgG1 isotype with less blunt junctions in favor of large homology ones, means there's a switch of reparation pathway in favor of AEJ1 from NHEJ. Our perspectives are to explain this difference seen on IgG1 isotype. Is it because this region is 4 to 5 times longer than those of switch alpha and switch gamma 3? So there's the recruitment of any specific nuclear factors to this region, or is it since it is the most reproduced isotypes? Or is it related to different subpopulations of B cells that some prefer switching using the AEJ pathway and some prefer to switch using the NHEJ1? We are planning to perform chip experiments investigating the proteins involved in each reparation pathway. So it's clear that the progress to come in the treatment of mature B-cell lymphomas will come from a better understanding of the mechanism leading to B-cell maturation, and particularly the 3' RR, since it seems to participate in both breaks and the reparation. And thank you for your attention.